Hello, Michael Lederman and Neil Greenspan here. We are editors of Pathogens and Immunity, and with us today is Dr. Ray Schinazi. He is the Francis Winship Walters Professor of Pediatrics and Director of the Laboratory of Biochemical Pharmacology at Emory University. He is the recipient of numerous awards and honors, including very recently an honorary doctorate from the Hebrew University Hadassah Medical School. Dr. Schinazi has a formidable and truly remarkable career in the development of effective drugs to treat viral infection. Among these are the antiretroviral agents Epivir, 3TC, Emtriva, FTC, and the anti-hepatitis C agent, sofosbuvir. An estimated 90% of the world's persons who are being treated for HIV infection are receiving a drug that was invented by Dr. Schinazi. These agents have helped to save millions of lives by prevention and treatment of infection. Welcome to Pathogens and Immunity, Ray. Let's get started.
Thank you very much for this lovely introduction.
I'd like to ask you, as we were discussing a moment ago, about your family background in several countries and how you believe your experiences in these other places influenced your subsequent intellectual development.
Yeah. Well, I was very fortunate to have both parents very pro-education when I was young, and they suffered a lot themselves and were able to give me one of the best education money could buy. It all started in Egypt, where I was born, a country I love. When I was about 9 years old, my mother was very sick. She caught an infection, which was a yeast infection, and there was no medication in Egypt. You know, some of the third world countries don't have access to medicines, unfortunately. Even today, that was back 1958, 59. So you can imagine trying to find a simple drug, an antifungal agent, was almost impossible in Egypt. So my father was fortunate to know a pilot on TWA who brought some mycostatin, it was called, from the United States, which is fungicide. For those of us who are more familiar with that name, it's available in every single lab in the United States without cell culture. And that drug actually saved my mother. And I was very impressed. I said, wow, this is fantastic. How can a simple drug like this can actually cure somebody? My mother went on to live to be 94 years old. So that tells you that she had a long life. Thanks to this drug, we were ready to bury her, frankly. And this small molecule was able to really destroy the yeast and make her whole again. And it left a very big impression on me. The other point in my life is, at that time, even at 9 years old, I spoke 3 languages fluently. And I think languages are very important because they make you develop differently. Every language is like being a different person. Knowing the culture of the country, speaking Arabic, speaking French, and speaking English. Later on in life, I studied Spanish at university and learned Spanish because my parents lived in Spain. So again, I started learning a 4th language. I think, as you know, when you know a language, you know the culture of the country, you can joke with people, you can understand them, you can understand the politics, you can understand their jokes, you can understand their food, and so on. So I think that's also helped my way of development, because it's not just learning about science, it's how to solve problems. And when you can solve problems with languages, understanding different culture, that really helps.
Well, I was going to follow up with what early influences steered you to science in general and antiviral pharmacology in particular, but you've certainly given one aspect of that answer already, although that was a fungal infection that you experienced in your family. Quite a powerful story. Were there any other early influences that turned you toward science and your particular niche in science?
Yes, my uncle, Andre Namias, is a very well-known herpes virologist. He came to the US in 1948 as a student. He couldn't get to medical school, so he went to Austin and studied English literature and other things. And then he went to do medicine at George Washington and became a pediatrician, did his fellowship at Boston, and then moved to Atlanta. I knew about herpes viruses when I was 10 years old. So most people didn't know about it, but I did, thanks to my uncle. But I wanted to be a bit like him. And he was a big influence in my life. And eventually I worked for him for a few years. And together we helped develop acyclovir in the early days with Barro's Welcome. So that actually was a great experience with Larry Corey and Andy Namias. they actually published in New England Journal of Medicine the first paper on the use of acyclovir for the treatment of herpes genitalis, and it's a fundamental paper. So I was somewhat involved in this, and I knew about it, and I want to learn more about how to conquer viruses, and that's where it started.
Do you have any strong interests apart from science?
It's a hobby of mine. I like fishing in general, fly fishing and other fishing. I'm not good at golf, so forget about that. So I think fishing is a great one. I try to, whenever I get on a boat, relax. That makes me very relaxed to be on a boat.
And Michael?
Sure. There's an emphasis nowadays in our schooling on STEM, on having students focus their entire educational career on science, technology, engineering, mathematics. Do you think that's a good approach, or do you think that a more balanced approach to secondary and college education helps in the development of a scientific career?
Well, I think when you're young, you have the opportunity to study all sort of things, from history to science and mathematics and English and literature and so on. Then you have to specialize. But I think it's important to become intense about science in order to excel in it, because there's so much competition right now. And you don't necessarily have to know all 4 or 5 different stems. You can develop many other things, but focusing on, say, chemistry, biology, it doesn't have to, engineering is great too. I think it's having this plethora of opportunities is what you need today to succeed, but it's not essential, because you will be normally exposed in life to other things.
After your postdoctoral fellowship, you moved to Emory. Now, why did you do that instead of joining pharma, which is where so many antimicrobial agents are developed?
Well, my postdoc, after leaving England, I came to the US and I worked with Bill Prusoff at Yale, who was basically the grandfather of antiviral agent. He's the guy who discovered hydroxyuridine, again, a herpes virus compound that works for herpes keratitis. And the winters, first of all, the winters in New Haven were not very favorable. They were very cold. So I did apply to some of the major companies at one stage. I remember asking Morris Hilleman, may he rest in peace, who was one of the giants in vaccinology, if he had a position at Merck in the field of antivirals. They said, well, at the time, they said, we don't do antivirals. Of course, now they do, but at the time, they didn't do any antivirals. So there was no position for me there. And pharma is also very much, how should I put it, they want people who are in boxes. And maybe things have changed now, but they wanted people who are chemists, stay in chemistry, and biology, stay in biology, virology, be virology. They don't intermix disciplines. On the contrary, I was a chemist who wanted to be a biologist, who wanted to become a virologist, who wanted to become a pharmacologist, who wanted to become a biochemist. And that's exactly what I did. So I was a bit out of the box at the time. And I felt academia for me would be best. So imagine, here's this young man coming from Yale to Emory, and I'm actually swabbing the vagina of women using a lab coat in a clinical lab. I'm organic chemist. How about that? And you learn a lot. You learn a lot because you actually see the disease for yourself with your 2 eyes. Like when HIV came along, I was actually in a clinic. My lab was next door to HIV patients, and you could see them outside the door dying slowly. And that leaves you a tremendous impact and motivates you to find a solution to the problem. When you have putting the clinical people with the PhDs side by side, this is so important.
And you point out an important value of being at a medical center where human beings are being cared for and doing research on ways to improve their lives. At pharma, you might be a little farther removed from that unless you've got some intimate connection with a medical program. Your approach to development and the invention of antiviral drugs, is it distinguishable from the approach that pharma typically uses, or is it similar methods but in a different environment?
I like rational design, but everybody else can do rational design. I'm a bit more a risk taker. So I use my knowledge and intuition to develop drugs. Pharma is much more rational in its approach. And unfortunately, that's not something I wanted to do.
So it sounds as though you were in a position to take some risks that pharma, who are often risk averse and somewhat complex in terms of the green light, might not be willing to do.
Yes, absolutely. They rarely take a risk because they have to go to their management and explain everything. It's what you find on the way to sometimes. You find unexpected things. Pharma would never have discovered FTC and 3TC, frankly. The whole story there is a good one. You have the idea of making oxothioline nucleosides. What is an oxothioline nucleoside? It's a nucleoside containing an oxygen and a sulfur attached to a carbon in a 5-membered ring. Now, any rational chemist will tell you that this is an impossible molecule to make. It will break down in seconds, including pharma. Okay? They will give you 10 reasons why it will not be stable and why it will break down. Well, guess what? We made that. I actually went to my mentor, my PhD mentor, and showed him the structure in confidence. Can we make that? And he said, no, that's my boss, my old boss. That didn't stop us because we went to a meeting in Montreal, an IAS meeting, I think it was at the time, and Professor Bellon showed some dioxylanes, 2 oxygen on a carbon, and that told me, wow, it's possible. They were all wrong. And I observed that. I said, well, if you can make double O, 2 O's in one 5-membered ring, why not the OS? And I came back home and we worked with some chemists and we were able to make similar compounds, but not lamivudine yet. We didn't make lamivudine or FTC yet, but there's also a bit of serendipity in science, as you know. So we were trying to make FTC, in other words, hook up to the base a sugar, the sugar with a 5-membered ring. And you do that with something called a Lewis acid. Okay. And turns out the postdoc was a chemist from Korea. His name was Woo-Bei Choi. And he didn't know anything about nucleoside chemistry, which actually helped, believe it or not. And he chose, there was the one bottle on the shelf in my lab containing stannate chloride, which is a Lewis acid. He decided to use that nobody uses stannate chloride in the business. But when he did that, he got a 99% excess of the right enantiomers. There are 4 possible enantiomers, the alpha and the betas, and then the basically racemates and enantiomers and the racemates, the left and right hand. And he was able to get exclusively 99% of the product was the beta derivatives, 2 of them. And the reason, I can go into the chemistry and of course the rationale, is tin chloride binds to the oxygen, has affinity for oxygen. So it hooks up from the bottom of the ring and forces the base to come from the top. Instead of making all 4 isomers, you only make the ones with the top one. Yes. How about that? And I won't go into more details because it's very technical, but it shows you what good science with a bit of intuition, with a bit of luck, can bring something very special. And then the next problem was separating the 2 enantiomers. And it turns out that one enantiomer is less toxic than the other. Both are actives. One is less toxic than the other. And one is FTC and the other one is 3TC that we made, and we actually licensed the process to, at the time, Glaxo, and they made 4 tons of lamivudine using our process. People don't know the story, but this is an amazing 4 tons. Imagine making, because of that lucky, they could not, with all their chemists, with all their resources, they were unable to make what we did at Emory University with my friend Dennis Liotta and Woo-Bei Choi.
That was a fantastic story.
There are stories to every one of my molecules. It's not an accident, it's a lot of hard work. In fact, one of the best stories is the story of the discovery of hepatitis C, and nobody knows it better than I do, despite other people claiming to have invented this molecule. They don't. There are basically 2 parts to the molecule. One is the drug itself, and the second part, of course, is the prodrug part. The prodrug part was well known. They used the McGuigan approach, but the story is a very good one because it involves some amazing people we hired at Pharmaset, the company I founded, that had a vision. We had a vision together, very little money, You don't need much money to make big discoveries, at least initially. And we were very lucky at the time to be able to buy a real-time PCR machine, which at the time cost about $500,000. So technology helped us. And then one other part was the discovery and development of replicon systems for hepatitis C by Ralph Batterschlager and Charlie Rice. These were critical. So having these two components, and the third component being people, qualified people who knew about virology, who knew about genetics of the virus, because we always assume that the virus is going to mutate and we'll have to counter that at some stage. We were able to find in our library a compound that worked against hepatitis C. It was actually a 2' fluoro cytidine nucleosides. Look at cytidine with a 2' fluoro, very simple molecule. But that compound was not potent, but it gave us a lead. And then we made the difluoro, which is gemcitabine. You probably know what that is. It's used as a cancer compound. Believe it or not, in humans, it does have an anti-HCV compound, but very weak inhibitor of HCV in humans. And we never published that, but we do have a patent file somewhere. And then we made the 2' fluoro 2' methyl. Now, this is the genius. This is the new molecule that had never been made before. It wasn't easy to make. It's a lot of hard work. Nobody thought about making that at the time. Like nobody thought about making FTC, and I can go why later if you wish. But clearly, this was a novel thing to put two derivatives at the 2' position. One of fluorine, and nobody put fluorine in his right mind because we had the experience of FMAU and FEAU. Remember these compounds for hepatitis B that killed 2 patients that Jack Fox and Kiyo Watanabe developed at Sloan Kettering and licensed them. And then Lilly failed miserably with these compounds. The drug failed, I should say, not them. But unfortunately, they killed some patients. So nobody in his right mind would put a fluoride 2' position. Well, we did, but we put it in the down position, not the up position. So there's 2 positions. There's what we call the ARA position, and then the ribose type of position. So we did that, and with the methyl group, and the compound was a cytidine analog, and the compound went all the way to the triphosphate, and the triphosphate inhibit the HCV polymerase. So we were clapping and opening a bottle of champagne. We had the first major hit at the company, and we actually licensed that compound to Roche because we always need a big partner to develop these drugs. But at the same time, we also discovered that there was a weakness to the compound. I discovered that actually. The weakness was that the compound can get deaminated at the monophosphate level, to the U analog. And we knew that from monkey work that the U analog is not active at all. It's totally inactive. But we also noted a brilliant biochemist by the name of Murakami, a Japanese scientist who worked at Pharmacet, worked out the mechanism of how this drug is metabolized in cells and found that the U analog, once it's formed, is actually phosphorylated. And the end product of this phosphorylation is the active ingredient of what we know today is sofosbuvir. It has a long half-life. That's why sofosbuvir can be given once a day. And it has a good affinity for the HCV polymerase, very selective for that. So then the obvious thing to do next, I shouldn't say that obvious, but it was obvious to make a prodrug of the U analog. So we had, and also we had a clue because we were never able to develop resistance to the cytosine analog. And the reason is quite simple now that we know all this, because we actually produced 2 drugs with the C actually became a U analog. So we had, not entirely, so we had a C plus a U in cell culture, and it's almost impossible to develop resistant virus to what we call 6130, PSI 6130. So the next step, of course, was to make the prodrug of the U analog, and that became sofosbuvir. And we used the McGuigan approach because the actual molecule we used was off patent. So we could use it, and we didn't have to pay any royalties to anybody. So that's where companies do that. and we made the prodrug, and that became sofosbuvir. Now, the data was presented at a meeting in Berlin by Ed Gain. You may know this gentleman, scientist from New Zealand. We went to New Zealand to do studies because they have very accelerated development for INDs, especially in the area of hepatitis. And it's a long way to go, but it worked out very well. He's a very well-known scientist now, thanks to this molecule. Everybody goes to him. He made his reputation with sofosbuvir. And he gave the talk. Actually, I couldn't even get into the room. It was packed in Berlin. And the data was shown, we get just on sofosbuvir alone, sofosbuvir plus interferon, we're getting 90+% cure rates, SVR 24, 12 and 24, of 90+% sustained virological response. I can tell you, we could hear a pin drop in that room. I was just outside, the door was open, and I could see what was happening. I was stunned. Everybody was anticipating this talk. We actually had a board meeting for the company that day, and it was called the board meeting. We were obviously quite happy with the results because nobody had ever seen anything like this, as potent as this and as effective as this. So it was a wonderful time, and we called ourselves the Perfectovir group. Perfectovir, which is the perfect drug. Of course, it's not that perfect because you really need to really bring the power of this molecule is especially apparent when you use it with an NS5A inhibitor. And the NS5A inhibitor came later. Of course, Bristol Myers Squibb had already one, a very good one, but unfortunately they didn't come to an agreement. So they had developed their own. There was delay in developing the combination, but eventually we had a combination which today we know as Harvoni or Epclusa. These are fabulous drugs that work pangenotypic, that work against every single genotype. And what is amazing is the need of this drug in a country like Egypt, because 1 in 5 people in Egypt is infected with hepatitis C. So here's this young man who was kicked out by Nasser from Egypt in 1962. And I'm in Hawaii. I got a call from the ambassador to Egypt asking me to come to Egypt immediately. This was before the drug was approved. It was 2011, December 2011. February 2012, I show up in Egypt for the first time in 47 years. And the people who greet me are the military people, the same people who kicked me out of Egypt and my family out of Egypt. And they know I'm Jewish.
Amazing.
And I basically talked to them. And of course, they want my drug. They want our drug. They didn't know how to approach Gilead and so on. But we found a way for Gilead to deliver the drug to Egypt for $1,000. They were ready to pay $3,000, by the way. But
Per patient.
I negotiated down to $1,000. So I helped them. And then, of course, they made their own generic formulation. That's what they were very interested in also. They have great chemists there. They actually manufacture the drug in Egypt, in Alexandria, the city that I was born in. They make sofosbuvir there, and they formed a whole company there, a whole structure. And now they deliver the drug for $60 a cure, not a treatment.
That's really amazing.
And President Sisi last year invited me to Egypt for a ExCom meeting that invited all the African leaders, all the African scientists to come to Egypt to discuss medical issue, but especially hepatitis C. And there he offered the drug free of charge to every single African country because he could make it for very, very, very inexpensively. So I raised my hat to him.
You bet.
Our own president, as you know, has just decided last year or actually earlier this year, to do the same thing. But it hasn't happened yet. There are more than 2 million people infected with hepatitis C in the United States. So Egypt has become from a red zone where they've cured more than 4 million Egyptians of hepatitis C and rising every day. it was a lot of suffering. Every single family was affected in one way or another. And the first time when I went there, as I told you, it's the military who invited me because they had a lot of their soldiers dying of hepatitis C, and they want to do something about it. I don't think many people are able to say that one year you go to Egypt and get an award, and then the next year you go to Israel and get another award, like I got the honorary degree. So I thought it was funny that I got involved in both countries. And I'm very pro-Egypt and I'm very pro-Israel. I want both of them to live peacefully forever, if that's possible. And I think peace through health is a very important concept that we need as scientists to push forward.
Well, Egypt should be proud of you, as we are in the United States, for your amazing contributions to global health.
You know, you've also thought about hepatitis B. Can you offer any thoughts about whether you think it's going to be possible to eradicate a virus like hepatitis B through your small molecule approach?
Let's put it this way. I think it's not going to be as easy as hepatitis C because there's two places where the virus can hide in the cell. One is the cccDNA, which is a latent form of the virus, and also the integrated DNA. So you have to tackle both of them, and also the general applications. There are actually three problems. The replication, I think we can handle with the drugs we currently have. And some of them, I was involved in the discovery of these compounds like lamivudine and lamivudine. FTC, unfortunately, is not being used for hepatitis B. But people do take it, especially when they take FTC for HIV. It also has an impact with tenofovir or tenofovir alafenamide. It does have an impact on hepatitis B, but nobody talks too much about that. But clearly, so we have the replication of the virus, we have the cccDNA. I think the CAMs.
For the audience, can you just define a CAM?
It's basically a compound that interferes with the capsid formation in one form or another. There are many mechanisms. It could be preventing the virus from getting into the nucleus by interfering with the capsid, or actually deforming or displacing the genome inside the capsid, or actually breaking it up. And making empty capsids too. So that's what there's several mechanisms. So these compounds are well known in the literature. They've been dismissed by some people as being ineffective, but I believe they're wrong, and will be proven to be wrong. We have now developed some quite remarkable CAMs. You'll hear more about them. Data has been presented at the EASL meeting, and more will be presented later on this year at AASLD in Boston. Clearly, these CAMs can give you about as much as 6 log drop in virus. Wow. Even at dose as low as 10 milligrams per kilogram in a human being. That's pretty impressive. None of the compounds can do that are out there, the CAMs that are out there. These are capsid effectors. And super potent, femtomolar, well, I would say picomolar in culture, but they're very active in humans. But most important, they just not only bring the virus load down, they also have an impact on the HBs antigen. And that is directly linked to the integrated hepatitis B in the genome of humans. So the more HBs antigen you have, the worse it is. So they're beginning to see a decline, and the decline is still going on even after 40 plus weeks of treatment, and the slopes are still going down. Unfortunately, the patient starts at 4 logs of HBs antigen, not 2 logs. If it was 2 logs, we would have probably reduced it to undetectable, but it will take a lot more time. We estimate it will take anywhere from 1.5 years to 2 years when you have as much HBs antigen to start off. Remember, with hepatitis B, you also have the immune system that's intact. So you can basically tackle both. The immune system is working in your favor, unless, of course, you're immunocompromised, plus the drugs that we have. And we usually use something like entecavir plus the new CAMs. One of them is called 184. That's the CAM I'm talking about that gives you tremendous response. Everybody wants the home run with hepatitis B. It's amazing because we had the home run with HCV. They thought it was easy. It's not easy. It was not easy at all. But they seem to think, well, we cured hepatitis C. Well, we can do the same thing for HIV. We can do the same thing for hepatitis B, but that's not the case. But going back to hepatitis B, I think if we continue treatment for, say, at least 1.5 years to 2 years, we'll have the HBs antigen to undetectable, at least with the current tools we have. The hope is that the patient will seroconvert at that point, and once it seroconverts, you have a cure.
When you give these CAMs, can you provide some estimate as to the turnover of infected cells in the liver?
Yeah, the infected cells, they used to think the infected cells, the turnover is very long, but people have done some work and they think the turnover is about 2 weeks. The problem is that you need 2 to 4 weeks, let's say. But even that, the problem is you got to make sure that the drug is constantly there, both the nucleoside or whatever you plan to use. It could be an immunological approach too. I'm not against that either, or siRNA approach or some other approach plus the CAMs. But the CAMs are going to be the driving force, a bit like what you have today with the nucleosides. The nucleosides, the driving force, they give you 2 log plus, and then for HIV, and then you use, of course, an integrase inhibitor or a capsid inhibitor with HIV. The new capsid inhibitor looked very promising, lenacapavir specifically. So I think it's going to be practically the same thing for hepatitis B. Now, I'm not saying that it's going to be a home run or a slam dunk. I'm very, very surprised. I think we'll get maybe rates initially of 30 to 50% cure rates with this material, and that's going to be incredible, because remember, there's 250 million people infected with hepatitis B. So I really think that the opportunity is there, and I think we'll fine tune and improve things with time. We'll find what works best, because if you can find 2 very powerful drugs that you can put together, I don't think we have anything like the CAMs today. So if you can find with a CAM putting something else that really works well, you may be able to reduce the cure time to, say, 6 months or a year.
Oh.
And that's what we're looking for down the road. But with the current status, the drugs that we have today, plus the CAMs we have today, I think it will take 1.5 years to 2 years to clear the HBs antigen. And once you have that, you will seroconvert. Remember also that spontaneous seroconversion. There are people putting interferon because they know with interferon treatment for 1 year, you get 5% cure rate, approximately.
In natural infection, some
Yeah,
natural
also.
people are cured spontaneously.
Correct. So you will get a small percentage. You don't really need a control group for these studies, in my opinion, because we can do a control group if you want. No. But I think we all know when you stop therapy, the big test is going to be when you stop therapy, we know invariably the virus comes back. So to me, a perfect cure would be you stop the therapy, forget about all the markers. It's helpful to have all these markers, or surrogate markers if they are surrogate. I don't know if they're quite surrogate yet, but nobody really knows. But we're going to learn a lot about the markers that are effective and prediction, who can be cured fast, who can be cured slowly, who can be cured, not cured at all. And we'll need some other methods. So I think a lot of lessons are going to be learned in the next year or 2 in terms of hepatitis B. Once we can cure some patients reliably, like 20% or 30% or 40%. Once we do that, I think, but again, as I said, everybody's looking for the home run. That's not going to happen. Let's be cautious here. And it's a much more difficult virus to treat than hepatitis C. And I think the ultimate one, of course, is HIV. That's
where I want to go next. So you're optimistic about having some impact on curing Hep B. Do you think that we'll be able to have, and what are your thoughts about the directions to get a scalable HIV cure strategy?
A cure is depending how you define it, but I think a functional cure is definitely possible for HIV. And if you completely eliminate the virus and the virus doesn't come back when you stop therapy, whatever type of therapy you have, you're dreaming. It's going to be very, very hard. It's like herpes, trying to find a cure for herpes viruses. It's not going to be impossible. It's even worse with herpes because the virus integrates into the nerve cells. Can't reach the virus. People have thought about using horseradish peroxidase and other things, that retrograde axonal transport and all that stuff to try and cure a herpes virus, but nobody's been successful. The only thing that cures herpes, by the way, is age. So the older you get, the less likely you have to have a recurrence. So that's the only advantage of getting old, I guess.
Good to hear there's something that improves with age.
Actually, bad for the virus, good for us. So I think it's going to be much more difficult. However, I'm optimistic. I hope somebody finds something, but I don't think it's going to be by traditional approaches. We probably don't have the answer yet because we don't quite understand all the reservoirs. We don't even understand where the virus hides. That's the biggest problem. Everybody's shooting in a barrel. So I think it's going to be very difficult, not impossible, but you have to be optimistic. Now, in terms of functional cure, I strongly believe it's possible. We already have, in a way, a functional cure with the Truvada and the Scovy and these type of drugs. We have that. I mean, it's a functional cure, whether you like it or not. You give the drug forever, you never get recurrences, and you're safe. Of course, you have to take the pills every day. Now, there are ways to improve on that, and people are working on that in terms of long-acting. But I think there are other ways of doing it. One possibility, I think, is with antibodies. I think antibodies are underrated today by a lot of people. Yeah. In particular, the drug companies. They will eventually start working on antibodies, I think. Antibodies are going to be very, very useful, provided you don't get antibodies to the antibodies. That's the biggest problem, as you know, the biggest challenge.
Useful in terms of their activity and duration of their activity in vivo, or useful in terms of providing a mechanism for eradication that's not provided by the current agents?
Well, it's not eradication. You're suppressing it. Still, virus still there, but you're suppressing it very effectively. And we've seen that with monkey experiments, where in neonates as well, in young monkeys as well as adult monkeys, where you can suppress the virus for a very long time. Unfortunately, I mean, I'm talking about years, as well as you can do it with Truvada. But the problem is, in some patients, in some cases with adult monkeys, it's actually only a small percentage of monkeys produce antibodies that are sustainable with sustainable levels. And that's the problem. And the reason is that you have antibodies formed against the antibody. If you can reduce that or eliminate that, I think we have a winner. And it's going to basically take over the whole market, in my opinion, of the current drugs that we have in patients. You can use it therapeutically, people already infected, as well as for prevention. So it's really wonderful from that perspective. And if you put it to say in an AAV vector,
right,
and you put the right cocktail of antibodies with the right conditions, you have an amazing opportunity. So that's something that I support fully, and getting more and more interested in destroying my antiviral legacy with antibodies.
So what's your favorite vector for getting into cellular production of antibodies?
Well, AAV, there are many types of AAV that you can have that target the liver. You give the liver intramuscularly, it's one shot, and that's it. And there's been evidence that these antibodies can basically persist a long time. In animal models. It hasn't been done in humans yet. So we have to see, for HIV, we have to see what happens in humans. A lot of animal work has been done in mice as well as monkeys. So I'd love to see that in humans. And I think there are people starting to do studies along those lines. So I'm very excited about that. I think the CRISPR stuff and what you hear of ARCUS, CRISPR, all that is a bit of science fiction. I'm not saying I'm not against it, but where are you going to go? Where are you going to go with that? You still have to deliver. So I think there is a lot of hurdles, but I encourage people to work on it. Hopefully it's not wasted time, but we're going to learn a lot.
Won't be wasted.
It's so refreshing to hear someone such as yourself give a serious discussion of the challenges of cure, as opposed to just throwing the word around loosely as if it's like inevitable that we're going to cure every disease, when it's been, as you yourself said, I mean, hepatitis C is an amazing achievement, but it's only possible because of certain features of the virus and its life cycle and so forth. And, you know, other viruses, other infectious agents present different kinds of challenges. And, you know, other than prior to the hepatitis C drugs, I mean, the only cures that were routine that I encountered were antibiotics for bacterial infections. I mean, there are very few chronic diseases that we cure. So that sets up my next question, which is, what about curative small molecule drugs or other approaches for, you know, other more common viruses that are not chronic viruses?
Well, I think the traditional discovery of small molecules for yellow fever, Nipah virus, whatever, is going to continue. The big issue I have is big pharma interested in this?
Right.
And because
short term,
practically, there's not much money for them to be made. And how do you get the industry to engage. I've seen Janssen do some wonderful work in Africa and with their TB work, for example, and also their new drugs on dengue. They look very, that was discovered with our colleagues at Leuven University. They've done an amazing job, but was that going to see the light of day? That's the question. Is there, so I think this is where I think NIH and BARDA and other organizations, even involving WHO and even small countries where these viruses are endemic, could play a big role with the help of American scientists and also European scientists and Chinese scientists and everybody else, but especially American scientists who are fairly advanced in this area and working in this area, encouraged by the NIH to develop drugs, for example, for yellow fever or Nipah and so on. What do they need to get together and set money aside for development of these molecules so we're ready when these viruses cross our border, we'll be able to have something to defend.
That looks more and more likely.
Yes.
Climate change. So
with global warming, already West Nile is a big problem in the United States where people don't talk about it, but there's over 1 million people getting infected. So I think it's coming.
Can you crystallize any unusual insights that you've had that have helped inform your career and lead to the successes you have? I mean, you've sort of hinted at several in terms of your comparison between your ability to be more free-ranging in an academic environment versus in a typical pharma environment.
I think it's not just me. As you know, these molecules are not invented by one person and are not created by one person. It's a lot of people involved. And the key is about people, quality people. We have to train more virologists. We have to educate them about drug discovery, drug development. We have to teach them about small molecules. We have to teach them about big molecules. We have to teach them about oligonucleotides and mechanisms. There's a lot of teaching to be done. And working in a lab like ours, young people can learn a lot, but it's very hard nowadays to find talented virologists, as you probably know. And we need to have a program to bring in more virologists to become interested in not only in drugs, but also vaccines.
You developed a lot of very successful molecules. What's your favorite antiviral that never made it and why?
I can think of 2 drugs immediately for HIV that I thought were going to be blockbusters. One was emtricitabline. You may be familiar with that molecule. It's a doxylin purine nucleoside that went all the way to phase 2 clinical trials. And unfortunately, Gilead, it was being developed by Triangle Pharmaceutical. Triangle got sold to Gilead. It sat on the shelf at Gilead for a year, and then after a lot of discussion, we returned to Emory University, and we tried to restart this, rekindle it. We spent millions of dollars developing it here in Atlanta with a small company that I had formed, and even had the clinical studies ready in Argentina to do a study. But unfortunately, we started the study, but we couldn't recruit enough patients because we had all these other wonderful drugs available. And we just had to throw in the towel. It was very sad because I knew that drug was as good as tenofovir with less problems and more potent than tenofovir. And it could have been a great addition to our armamentarium against all these HIV drugs. The other one was a drug that we licensed to Incyte, believe it or not, at time when they were getting away from all that gene sequencing activities that they were doing before the JAK inhibitors came along with Incyte. And they licensed it and they did clinical studies, but this compound was probably the most potent nucleoside at the time on the planet. We had done a study in humans. It was a PK study, but they infected patients. And after one single dose, we had almost half a log drop. It was actually interesting. Most people don't go around testing drugs after one day, but I did. It was the first time. A lot of people are doing it now, but at the time it was totally novel. This is maybe a creative way of thinking. Why not? If it's going to be so powerful, we know the virus load comes down very fast. And we showed a reliable half a log drop in all the patients. It was a PK study, but we had saved the plasma, and we were able to measure the viral load drop. And And we've actually published that paper. And as I said, a lot of companies have copied us now because you don't need to do long-term studies. You can find out very quickly whether the compound is going to work in the clinic or not, or even after a single day. So that compound was so powerful, and I think it was completely mishandled. The combination they selected was not the right one. They had some of the old drugs available, nevirapine and things like that, they put together with our nucleoside. And eventually we found out the company was not really interested in antivirals. They went on to the JAK inhibitor, but I still think it's a wonderful compound. But nowadays we probably don't need, we have a lot of very good drugs for HIV.
JAK inhibitors have demonstrated efficacy in a number of settings. Do you think there are other or any host-targeted approaches for prevention of HIV infection that will turn out to be useful? You're optimistic about the the production of antibodies in vivo, but are there agents that you think will target host elements that the virus needs for propagation?
A lot of people are working in this area, and we're discovering new host factors that are important for HIV and other viruses. Recently, we saw some papers on sphingolipids and that type of enzymatic mechanisms that are involved in lipid metabolism and so on. So I think they're not targeted specifically at the virus. So I think it's great. There's also people who are in the contraception area that are looking for compounds they could use in combination with the current contraception devices that we have to slowly release to prevent STDs, including HIV. And they're looking for polyanion compounds that could be effective, algae type of extracts and so on, that are charged molecule that could affect the receptor to the virus. So I'm all in favor of that, but my personal career has been focused on mechanism. So that's what we try to find out, what the mechanism and confirming the mechanism. Obviously, to me, primarily the mechanism in the case of HIV has been the polymerase. For hepatitis B, my career has been for HIV and for HCV and for HBV has also been polymerase, but we also look at other targets that could be useful, like the CAMs I mentioned earlier.
Have you thought at all about mRNA-based therapies?
I think that's an evolving field and very exciting. The big question, of course, is trying to, we've seen the success of the COVID vaccine with mRNA. They're not perfect vaccines, but they certainly did their job as best as they could. They're not durable, unfortunately. So I think there's a lot of room for a lot of improvements. But again, they went very fast, remarkably fast. It's been very successful. It's a proof of principle that they work, and now we can now apply them to a lot of exotic viruses, a lot of exotic diseases, and even not so rare diseases as well. It's an evolving area, and the numerous companies that have now started up all over the world, especially in the US, even one in Atlanta, believe it or not, that is making these mRNA. They have to be tested, of course, in animals to demonstrate that they work both therapeutically as well as a prevention. And they could be extremely, extremely good. So I'm all in favor of that. And I think if we better understand what's needed, the requirements needed in terms of expressing the full length RNA or partial, what are the important parts of the RNA that are essential for its effectiveness and durability, that could be very, very, very good.
Yeah, I was thinking not only vaccines, but also actually encoding other sorts of proteins that inside the cell can interrupt viral,
Correct.
cellular processes necessary for the virus, whether they're host or virus encoded.
I think we're going to see a lot more of that at meetings. It's going to be very, very exciting. And it's certainly a new area, a refreshing area, because we need some changes. We can't keep doing the same thing forever.
If a physician is serious about a research career, what do you say about the need for recommendations for formal training in science, getting a PhD?
If they don't want to do a PhD, that's fine with me. I think the key is having protected time to do research. That's the biggest problem. I've been in this business for more than 45 years. We have MDs in my group, embedded in our group, they don't necessarily have PhDs, but they behave like PhDs, and they're also surrounded by PhDs, which will help them. And I think that's the goal. When you get an MD PhD student in your lab, you feel honored that they chose your lab. You know that these people are motivated, and you know that they are going to spend more time to get their degrees, and they will work very, very hard for you and for themselves as well. But I've seen MDs excelling in our labs in the past and going on to do big things.
I have just 2 more questions for you. Who are your favorite teams in Premier League and Serie A?
Well, I like Liverpool, and
Okay.
I think you know why.
No, you must tell me why you like Liverpool.
Mohamed Salah,
Okay,
Egyptian.
all right.
He's the pharaoh. He's the Egyptian pharaoh.
Well, he's amazing.
He's amazing. He's changed Liverpool has come from nowhere. And I think that's a wonderful story to show an African coming to England and being successful. In fact, a lot of Africans have done an amazing job. Mbappe in France, and it's remarkable the evolution that's taken place in soccer, football. And I'm happy to see that happening. It's a lot of fun, and it's great to see these young men playing beautifully. And with such talent and such speed. I wish we could have him on my team discovering drugs for HIV and so on. He's a fast runner, very fast runner.
You bet, you bet. Well, Ray, thank you so much. It's been a thrill for me, and I'm sure for Neil, to have you talk to us for Pathogens and Immunity.